we could just chat more, and because we probably need to wait a couple of more minutes anyway. So
well, we can wait if you like.
No, just open it, please.
Okay.
Yeah, because I mean, making people wait, outside while watching the whatever circling, just keep going. I hate that one, because we should let them, join as soon as possible, because whenever I kind of see those circling, I just, okay, I'll go restroom or turn off Zoom and probably not joining. So yeah. Oh, we have some chat going on. That's great.
Yeah, just saying hi. Yeah.
So I mean, today is kind of interesting time zone. I think Theresa, you are the Californian time zone, right? And Boston and Central. And next week, actually, we will have Australia.
Oh.
Wow.
Yeah, and then I think his time is 1 o'clock in the morning.
Whoa.
And yeah, so crazy. But last week was also Korea, and that was midnight. And Professor Sang Hyun-Lee actually stayed until the 1 o'clock his time, and then even gave some feedback after the young speakers talk. So I'm so grateful, because that is the reason I basically continue during the preparation period, because I overwhelmed by the excitement and all the emails I got. And then
I'm going to open the webinar. I'm sorry, that's just me.
Okay. Yeah, you could talk anything. You're my superhero, so don't worry. Yeah, so that excitement basically drive me until today. So I'm so excited. I mean, every day because of this seminar series. So any additional question? Anything? And you are ready to go?
Ready when you are.
Sure. Okay, so I'll give 1 more minute. People joining, and typically Zoom, because I have lots of Zoom meeting, and then Zoom meeting sometimes go over. So and then also switching, like typically 5 minutes late, I expect every time I kind of do Zoom meeting.
Yeah.
And interestingly, my school, when we deliver lecture by Zoom, we used to have the 10-minute in between the class, but during the Zoom time we have the 15-minute. I don't know why, because I mean, to me, I mean, in person and then going from one classroom the other one takes more time, but they kind of think additional 5 minutes should have some technical challenge, internet problem somehow. So that's how my university basically implement for last semester, actually for 1 year actually. And now
back to teaching in person now, or are you still teaching?
back to and my university is super great in terms of making sure the safety of the student. So we install all the filter, and then all the kind of air filter type of thing, also some machine that keep kind of making the classroom fresh. Yeah. So we invest a lot of money, and then I'm really grateful about those kind of investment. And then of course the safety of the student is #1 priority of the school. So we achieve a lot of great thing. I mean, so still okay, but we don't know. I mean, we cannot predict anything. So unfortunately, in this time. Okay, so I guess I should start. Okay, so let's start. So good morning, afternoon, evening, and hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요. And hello, everyone. Welcome and thank you for joining us the first seminar and Nature Chem Bio Day. Our rising star speaker, Theresa, just published her paper in Nature Chemical Biology, March this year. And we will have the keynote speaker, of course, Caitlin Deane, an editor of the same high impact journal. So I call it, Nature Chem Bio Day today. It's my absolutely tremendous honor to introduce very briefly our keynote speaker, Caitlin Deane. Well, what else do you want to know about her other than she is an editor of the journal to which we hope to submit our paper, right? So instead of introduction, I just want to thank her for her kind offering. She will be available after this seminar. I mean, after we finish Q&A around 11 a.m. my time, that is the Central US time, and we'll meet again via another Zoom link, which I will provide on the chat box later. So I believe, from Dustin, a magician and a brilliant storyteller at America's Got Talent, and she basically got the $1 million yesterday. And I wanted to use some quote, what he said. So quote, "Giving is the foundation of change," unquote. I want to say we are all busy, but we can change this world together by giving your time, just 5 minutes, resources, love, attention, support, kind words, and forgiveness. Even if Caitlin gave me, sadly, a rejection letter, it was helpful to move on to the next journal without a long delay. Of course, I provided my expertise and time to review for her journal multiple times. So young people, if she asks for your help to review papers, please give your time with carefully gathered constructive feedback rather than rejection or acceptance recommendation without careful review. Okay, I guess rather a longer introduction than I expected. So Caitlin, the virtual floor, all yours.
Thank you. Yeah, thank you for inviting me to do this. My background when I was in school, doing my graduate work, was not really synthetic biology, and so I don't necessarily think of myself as a pioneer in this field. But so I'm really honored that you invited me to give a little introduction here. And actually, the quote you just gave about how giving is how we affect change in the world is really kind of what I wanted to talk about, because I think, thinking about synthetic biology as a whole, what I really appreciate about this field is that everything is very focused on how do we make tools that improve the world? How do we make tools that other people can use? There's really this spirit of generosity in the field that looking back, I really appreciate, because all of you who are doing this research are always so focused on how can we make these tools useful to other people, and how can we enable these biological insights that other people want to find. And that is what really makes synthetic biology, I think, a core part of chemical biology as a whole. Looking back, there were synthetic biology papers being published in the very first issues of the journal in 2005. It's been part of the field as we've seen it from the beginning. Because I mean, the central idea of chemical biology is how do we look at these biological systems on a chemical level? And I mean, that's really what synthetic biology is doing, is you're breaking down these complicated problems into their component parts and building them back up again. And you can't build them back up again unless you really understand what's going on. And so that's what really makes synthetic biology chemical biology as well. And yeah, so the idea is, how do you make, you're making biology work for you by building these tools and looking at chemical biology, what we've published at the journal over the years, and I think ever more so in this way, the synthetic biology is touching on so many other areas, whether it's developing the genetic tools that are necessary to activate biosynthetic gene clusters, or facilitate the metabolic engineering types of studies that can biologically produce useful compounds, or the genome editing that's used by everybody these days, or the protein engineering methods that have enabled us to have fluorescent proteins and optogenetic tools, biosensors, protein-based biomaterials. I mean, these are all things that build on synthetic biology tools. And so that is really what, going back to that core idea of, I really appreciate how synthetic biology is so giving of these tools to the greater community, and this spirit of interdisciplinary studies, and this spirit of generosity and collaboration that really goes throughout the field. And I just really appreciate that. So we're all here to hear Theresa talk, so I'm gonna hand it over to her quickly, and I'm gonna let her tell you about her own project. But I do wanna note that because we had the honor of publishing her paper, I looked it up this morning, and right there in the abstract, there is the phrase user chosen, I think, or user defined, or something like that. And so user selected, excuse me, user selected. And so the spirit of making things easier for the user and making tools that are useful to other people is right there in Theresa's work as well. So with that, I'll hand it over, hand it back.
Thank you so much. That's brilliant talk. And I really love what you said about your journal, and then your kind of perspective and your vision, and that's all I wanted to hear today. And also, I also realized, I also know, that one before, she cared for synthetic biology. That means I encourage young people to submit your papers to her, and then she will consider very seriously. Okay, so now the main speaker of today. So I will just give a little bit longer introduction. So it was so easy to select Theresa as our fourth speaker because, as I said to you earlier, she published her hot paper this year in Nature ChemBio, of course, and and then that paper is kind of very, very interesting thing, and then she probably talk about a little bit about that paper, also probably a little bit more new data today. And to give us some idea about her, she received her PhD in cell biology from UC San Francisco, where she studied DNA damage, the cell cycle, and the control of protein levels by the ubiquitin proteasome system. As a postdoctoral researcher in the laboratory of Chang Nu, I believe genius scientist and engineer in the Department of Biomedical Engineering at UC Irvine, she has created a DNA recorder that basically works with the host DNA damage response to encode the lineage and stimulus history of mammalian cell. Today, she will present her most recent postdoctoral work, and that's kind of upgrade version of the recorder I just mentioned, a much more faithful and durable recorder that opens up the possibility of recording many inputs over weeks to months. Theresa is currently applying for faculty positions with the hope of developing the same recorder into a tool that records the activation history of many signaling pathways in parallel in physiological settings over the whole timescale of developmental processes. She is a leading edge fellow and Mosaic K99R00 scholar. So if any hiring committee chairs are in this Zoom room, do not make her choose the other schools by your late response to her application. Okay, Theresa, take it away.
Well, thank you so much for that really, really kind introduction. I'm completely honored to be able to present in this really wonderful seminar series that's, I think, bringing together a worldwide community of synthetic biologists. And Caitlin, thank you so much for talking and sharing your thoughts. You sort of, I was feeling very inspired because what you described with this idea that synthetic biology is really about giving, right? About making tools that are actually ready to be used by other people. I mean, that's sort of my goal that I hope I can reach with my work, and kind of the underlying value that I really want to make central to my future lab of really trying to give biologists and other synthetic biologists tools to really do the experiments that they find most fascinating, and kind of really expand the universe of what's possible in our field. I would be very happy if I could do that sometimes. So today I'm going to talk to you about how we make tools out of proteins and nucleic acids that can change DNA, and how we sort of take those tools and put them in cells with the ultimate goal of following what's going on in those cells as they develop into tissues or organs or whole organisms. So backing way up, I think we can all agree that DNA is durable and heritable, and that therefore, if you make a change to DNA, that's also going to be durable and heritable. DNA recorders are synthetic biology tools that take advantage of this property of DNA to convert a transient event into a cell into a durable change in its DNA that then can be read out later in the experiment. And this is a really exciting and sort of burgeoning field. So I just wanted to shout out all of this great work done by others that, in part inspired me to get into the field, even though I don't have time to go over everything in detail, of course. And, I'm sure many of you are already familiar with this work. So the sort of metaphor that I like best for DNA recording is that of a flight recorder, because the idea is that you make the recorder sort of keep track of the parameters in which you're most interested. And then you sort of load up the cell with the recorder at the beginning of the experiment and let it fly and make an organism. And then at the end of the experiment, you can extract the recorder from each of the cells and read out what happened in that cell's history. So I think in order to really understand kind of some of the features of DNA recorders that I'm trying to develop, it's instructive to think first about the simplest possible DNA recorder. And that's one in which a single event in the cell leads to a single change in a cell's DNA. So perhaps the event leads to the expression of a DNA modifying protein, which leads to this durable change in the DNA. And that any cell whose forebears underwent that event will sort of be apparent at the end of the experiment because it will have this change in DNA. So you might see that I've made these changed cells green because one of the sort of most common sort of original DNA recorders are, something like Cree-lock systems that tag every cell in a particular lineage. So the way this works, a little bit more cellular detail, is that obviously cells start from very few in the course of at least mammalian development and all multicellular organism development. And they divide and proliferate and make many different cell types. So imagine this one cell at this stage of development underwent an event that we're trying to record. And so all the different cell types that arise from that cell are going to be marked. But in order to really study this process or the developmental processes in which we're interested in more detail, of course, there's sort of two extra things that we want to be able to capture. The first is that there's obviously not just this one lineage. There are many different lineages in this organism, and it would be really advantageous for many different biological questions if we could keep track of all of the lineages. In addition, of course, there isn't just a single point in development at which interesting and important lineage specifications happen. So it would be really advantageous if we could keep track of multiple successive differentiation events throughout development. So one kind of feature of DNA that can be used to sort of expand the breadth of events that we can keep track of is that you can set up a system where the existence of any mutation indicates that some event in which you're interested took place. But if there's some variation in the exact nature of the mutation that happens, that those different mutations can help you keep track of different lineages in parallel. And so this is something I'm going to come back to. But first, I want to talk about the other half of my kind of introduction to DNA recording, which is what's a biological question for which this technology is sorely needed. So I think the clearest use case with which I'm familiar is that of really trying to understand metastasis in cancer. So in order for a primary tumor to generate metastases, you have to have some number of cells that start to migrate away from the tumor, invade the basement membrane, intravasate into the circulation, survive in the circulation until they're able to extravasate, find a niche in which they can start proliferating again, grow a small tumor, and eventually grow a large tumor. And this process is, of course, an important biological process that can be a window into many sort of basic biology questions, but is also a devastating medical problem that there's really an urgent need to understand how this happens so that we can stop it from happening for real patients. So how do we want to kind of understand what made this cell do all of these different fate changes so it could become a metastasis, so that hopefully eventually we can stop it from doing that? So one thing you might potentially want to do is to really study the cells that have actually formed a metastasis, perhaps in an animal model, in great detail. And there are many techniques by which we can do this now. For example, you can take these metastases and do something like single cell RNA-seq, and really, to understand the full transcriptome of the cells and really try to understand in detail sort of what their phenotype is at this point in the process. However, by the time the cell has actually grown a metastasis, it's not necessarily exhibiting the same features as it did, for example, when it was able to extravasate from the circulation. And, it would be much nicer to stop it as early as possible. So then you might imagine, okay, well, let's find a model in which we can really capture those cells that, are in the process of, doing one of these earlier steps to understand what they're doing. The issue is that this particular cell that's able to extravasate, we know, because this is a cartoon, that this is the cell that's going to go on and form a metastasis. But in fact, there are probably many, many cells that are able to get to each earlier stage of the process, but are not actually competent to form a metastasis, and instead will be, for example, killed by the immune system before they can cause any problems. So what you really want to do is to allow the cells to do what they're going to do and say, okay, we know that all these cells that have grown a metastasis were competent to grow a metastasis, but be able to look at them at this point and see what they were doing at all of these earlier points in their fate change from, a member of a primary tumor into a metastatic initiating cell. So this is why we want to make DNA recorders, so that we can keep track of the history of a particular cell while it's in its physiological context. And therefore, at the end, we both know what phenotype it ended up with, and we can read out its history. So if we want to make a recorder that's capable of doing that, what do we want in it? I think we want it to be high information, so we can keep track of many parameters and many different lineages. We want it to record in order, if possible, both so that we can reconstruct the order in which different events happened, and also because even for lineage reconstruction, recording in order greatly simplifies the lineage reconstruction process, sort of mathematically. We, of course, want the recording to be able to last over the course of the whole experiment or physiological process in which we're interested in. And we want the recording to be not corruptible. So basically, if you've recorded something, you don't want to come in the next round and erase what you just recorded, because you need to be able to read out the whole thing at the end. So first, I want to talk to you about my published work, which was my first attempt to make the sort of nuts and bolts of a recorder that would sort of have some of these parameters that people could use in their larger recording systems. So Chiron takes advantage of a previously published technology called a self-targeting guide RNA. And what you need to know about that is that, as we know, Cas9 combines with a guide RNA in order to target a matching DNA sequence. And then it cuts the DNA. And the trick with the self-targeting guide is that the DNA that's actually cut is the same DNA that encodes the guide. So instead of having the DNA be cut and mutated, and then it's game over, you'll come back around and get a new guide that can then combine with Cas9 and lead to the next round of cutting of the DNA. And my innovation was to combine this with a mammalian protein called TDT, which, when Cas9 makes this cut, it makes a probably mostly blunt-ended double-strand break, and TDT is a non-template dependent polymerase, which will bind to that double-strand break by interacting with the native DNA repair, and actually convert this break into an insertion mutation. And because of the exact geometry with which Cas9 cuts, those insertion mutations can be accumulated in multiple rounds, and they'll be accumulated in the order in which they were added temporally. So let's talk about the parameters of chiron from my Nature Chemical Biology paper in keeping with Nature Chemical Biology Day. So this is, we made a cell line that has chiron locus, which is just a self-targeting guide integrated into the genome. And over time, this is the average length of insertions. So you can see that the length of insertions is getting longer as the cells are recording over time. And ultimately, we average about 14.6 bits of information. So that can distinguish many, many different lineages, for example. We showed in the paper that this recording does indeed happen in order, which I don't have time to get into the raw data, but basically this is just due to the architecture of where Cas9 cuts and how the self-targeting guide works. And the recording is moderately long lasting. So we're continuing to accumulate edits through a week and a half, which is a timeline that covers many interesting developmental processes such as wound healing. And here you can look at it in a different way, where we're just looking at what kind of mutation is at the chiron locus. So we start out 100% of the cells have the original root sequence, and then that declines over time as we're accumulating insertion mutations, as we're continuing to accumulate them over time, but also deletion mutations, which I'll talk about in a few minutes. Given these, really quite good parameters, we decided to test whether chiron could actually faithfully record events of interest. So here we're reconstructing the relatedness of different populations of cells in a setting where we sort of know the relationships because we split the cells in this pattern to end up with 16 different cell populations and I think what we really needed the chiron parameters for is that there's over 3 million cells per well. And basically we sequenced the chiron locus just from the final cells, and we were able to accurately reconstruct all of the relationships between the cells. And in fact, we had way more information than we needed. So we could downsample it quite a bit and still get perfect reconstruction. So next, we looking at these parameters, we thought, okay, so as time goes on, you're accumulating more insertion mutations and therefore a longer insertion. And so if you, for example, tie the chiron recording to a stimulus of interest, you should be able to get longer insertions the longer the cells are exposed to the stimulus. And we did this with a chemical that activates the hypoxia signaling response. And indeed, we found that just by looking at the average insertion length in the cells, you could accurately reconstruct sort of the relative times that they had been in the hypoxia mimicking compound. So looking a bit more closely at some of the ways in which we still want to improve this technology. So the rate of sorry, I can't see something, but anyway, the rate at which the chiron locus is corrupted is much lower than if you were just cutting with Cas9, where you would almost always get deletion mutations, but it's certainly not zero. So you can see that as we're accumulating insertion mutations over time, we're continuing to get insertions as we would expect, but we're also having these deletion mutations pop up, and those represent a loss in general of the whole locus, essentially. So that's because although in each round of recording, you're much more likely to get an insertion mutation, as you accumulate more rounds, each time you go through a round, you have about a 25% chance of getting a deletion. And as deletions are sort of a lowest energy state where nothing else can happen, as your number of rounds goes to infinity, obviously your uncorrupted loci goes to 0. And in fact, we get an average of about 3 rounds. So if we look at this from what I told you, where we have this mix of insertion and deletion mutations, here's just a single round of recording to show you that indeed we have about 75% insertions, but this rate of deletions, which we were not able to depress further, is eventually, in the limit of many rounds of recording, is eventually going to corrupt your loci. However, because all of these parameters were quite good, we're now able to apply chiron in many biological systems. So we've made mouse ES cells with several chiron loci, and in collaboration, we're going to use it to really record lineage in some open questions in wound healing. And in addition, we want to use it to record cell lineage in a model of cancer growth and metastasis in xenografts. So what's the next step? I see myself as sort of someone who makes the smallest parts of DNA recorders and then hopes to make them into a working recorder for the use of developmental biologists, and some use that I want to do in my own lab as well. And I really wanted to make a recorder that would be truly long lasting and truly much less corruptible. So before I get to exactly how I think we've started to do that, I want to talk about where we were at this phase in sort of the evolution of the project, which was in late 2019. And at this point, I was really trying to push against where we were with genome editing technology, which is that if you wanted to have high information recording, that entailed making a double strand break, essentially. So obviously it's still possible to do a ton of really interesting both recorder making and biological experiments. But in order to really improve the basic technology, I was really stymied by the fact that, if you make a double strand break with Cas9 in the presence or absence of TDT, there's many possible mutations that can result. And so, for example, you can distinguish many different lineages, from just a single site of cutting. But of course, that requires making a double strand break, which means that there's probably always going to be a significant chance of getting a deletion mutation that will corrupt your locus. And also, although cells experience many double strand breaks in the course of normal physiology, if I had a choice about how I wanted to treat the cells that I'm trying to study without perturbing their normal development, I would not make double strand breaks. So on the other hand, of course, there are many really great genome editing technologies, and indeed the original DNA recorders, such as recombinase, those are much less damaging and, for example, maybe only make a nick, but there are fewer possible sort of resolutions of this editing event. And therefore, you need many more sites in order to do recording, which is sort of challenging, for example, for ordered recording. So at this point, there was sort of this trade-off, and I was really trying to think of ways to kind of push each parameter to try to push through this trade-off. And this is why I really love being in the DNA recording field, because it's both a really exciting field on its own, but also we get to take advantage of the extremely wide interest in making new genome editing technologies. So here are some ways. First, I want to call out some ways that I'm not using, but that I think other people are going to use to great effect to sort of abolish this trade-off. So first, and this was even older, in bacteria, CRISPR integrases are also kind of ancient DNA recorder that keep track of past events in a cell's life. And you can do really beautiful recording in bacteria with them. And I think that there's been work going forward that will, make these really flexible technologies for recording, and also potentially, move them into mammalian cells. And I think that that's going to be really exciting. Another way that came out in 2021 to try to abolish this trade-off is, or perhaps 2020. I'm sorry if I'm getting that wrong, but is to really make base editors have more potential resolutions of their editing events, which I think is also going to be really great for recording applications. So the paper that came out in 2019 that I'm going to take advantage of is this work from Anzalone et al. In David Liu's lab, where they made Prime Editor, which with just a nick can actually introduce a wide variety of mutations. And so I really, as soon as this paper came out, I really wanted to figure out a way to turn it into a progressive DNA recorder. So what you need to know about Prime Editor for the purposes of this talk is that it's basically Cas9 fused to a reverse transcriptase. And what it does is make a nick in the non-target strand of the targeted DNA. And the 5' end of the guide RNA is just a normal Cas9 guide, but the 3' end is this extension that actually acts as a template for the reverse transcriptase. So as a primer binding sequence, and then a template for the reverse transcriptase to actually install new mutations. And for our purposes, so Prime Editor can make many different types of mutations. For our purposes, the one that was most important is that it can make just a single strand nick, no bottom strand nick or anything is required, and make a moderately sized insertion mutation that's encoded by the guide. And our innovation was to figure out a way to make this moderate insertion mutation activity happen sort of continuously and progressively for the purposes of DNA recording. So this is how Prime Editor chiron, or PE chiron, works. So you have a DNA sequence that has this kind of gold target site in the middle. You have a pegRNA that directs Prime Editor to this DNA sequence. And then in its 3' end, it encodes both a primer binding sequence, a variable region that is here in pink, and that's going to constitute the record, and then the next target site for the next round of recording. So the idea is basically that here you make this insertion mutation, here's this pink record that's never going to be touched again, and then here is the next target site. And in the process, you've destroyed the first target site. So in the next round of recording, you have perhaps a similar pegRNA that can target this next site, but perhaps it has a different variable region that then is going to be incorporated into this recording locus. And you can do this in many rounds and so on and so forth. So by its very design, this recording can be high information because we basically have control over what mutations we make because we put them in the pegRNA, and it's ordered just by its design because there's at each point only the very last added site is actually available for editing. But the question is, what can we do to make it, or rather, is it long-lasting and incorruptible, and what can we do with it? So we were actually, we made a stable cell line that expresses all the PE chiron components, including 42 different pegRNAs, which are similar to each other, but in that they can do the progressive recording, but have different signature sequences. And so here, this is the process of adding new records. And here we're looking at the actual experiment in cells. And from reading out the sequences at the loci at the end of the experiment, we get a certain amount of each length. And basically, we can predict, based on the sort of the rate of the initial round of recording, we can predict how much of each longer length we expect to see. And we find that our actual observed efficiency is very close to 1 for at least these first 5 rounds, which really suggests to me that we're not losing efficiency or activity as we go over time, and that therefore PE chiron has the potential to do quite long-lasting recording and continue to accumulate these records. And just to mention kind of what was behind this, basically this particular activity of prime editor seems to be very sensitive to the exact insertion sequence. But of course, if the outcome of the event you're trying to screen for is a durable change in DNA, it's very easy to screen a very large library of potential sequences. So we screened about 100,000 and we got 2 that worked well. And we were very lucky that in the original paper, there was a sequence that they tried that happened to work very well. So that sort of enabled us to get our foot in the door. But I think the really important feature of PE chiron is that the signaling has very low corruptibility. So here we're not looking at rates, we're just looking at the total number of reads at the end of the experiment that have sequences of the corresponding number of rounds. So it goes down over time because the cells just haven't had as long to get to 5 steps. But here in pink at the top, we're looking at the correct edits. So they'll have a sort of recording sequence or a variable sequence recorded at random, but then also they're considered correct if they have the appropriate length and the appropriate target site for the next step added. And in black are all of the off pathway results that are a similar length, but don't have a record that's exactly what we expected. So you can see that for almost every length, we're getting at least 2 orders of magnitude fewer off pathway results than on pathway. Here, I think we had a particular sequence hole that the edits fell into that we're working on changing. This is just, but still, this is less than 10% of the edits of this particular length were the incorrect ones. And so I think this is huge. I think for our purposes of trying to get a really long-term recording, having something that almost always makes the right edit is essential. And that's the part of PE chiron that I'm most excited about for the sort of applications that we're aiming at. So given these good parameters, we tested whether recording with PE chiron could accurately reconstruct this sort of relationship between cell populations for which we know the ground truth. And in fact, the reconstruction was perfectly accurate, just as it was for chiron. So what are the next steps? So right now we're trying to move this into cell lines beyond 293T and actually into animal models of metastasis. But there's one feature of PE chiron that I want to call out, because each of these sort of different but mostly similar PEG RNAs that can do successive rounds of recording but have different durable recorded sequences, each of these is just encoded by a small PEG RNA. So if you could tie each of these different PEG RNAs to different promoters, for example, in signaling pathways of interest, you could potentially start to really multiplex many different records, and they would be recorded in order. And you could actually start to make that quantitative by having multiple recording sites in the cell. So for example, there are many line 1 repeats in human cells that have sequences that can be targeted by a single guide RNA, for example. So future plans. I really want to develop this technology and make this into something that can record signals in great breadth. I have a long-running collaboration with a cancer biologist at UCI, Olga Razornova, and we're really excited about trying to understand some of the pathways that underlie metastasis. And in my own lab, sort of my original scientific identity is as a DNA damage person, and I'm really excited about figuring out how the sort of physiological endogenous DNA damage affects cell fate, which hasn't been possible before. So I really need to acknowledge the whole Lou Lab at UCI, which has been a fantastic place to do science, and especially all of these people contributed directly to the work. I especially want to call out Cheng Liu, has been a fantastic mentor in every way, and encourage anyone looking for a postdoc or any position to come to our lab to apply. And Courtney Carlson is a really brilliant senior PhD student who has a lot of her own work that I hope will be visible soon about engineering TDT, and also has been my partner and co-conspirator in all things PE chiron. And here's all our outside the lab collaborators as well. I've really benefited by being part of the Leading Edge Symposium. And here's our grants, and I'll take questions.
Wow, fantastic, Theresa.
Oh, thank you so much.
Yeah, thanks for you the wonderful talk. I mean, I actually wanted to ask some question, but I want to check first whether we have, okay. Oh, we have one question. So the question is from Rosanna Molina. The question is, how do you do lineage reconstructions? When you say you reconstructed it perfectly, were there other potential solutions to the reconstruction, or were you 100% certain?
Oh, that's an excellent question. So I should also, for full disclosure, Rosanna Molina is this person. She's in our lab. What we were doing there is reconstructing the relationships between populations of cells. So the Elowitz lab has done some really good work where they actually can do single cell level reconstruction where they know the ground truth because they've been watching the cells continuously. What we did here was reconstruct the relationships between populations, which we do know exactly how related they are because I split them in tissue culture at times that I know. And so I know what their relationship is to each other, essentially. But going forward, as we want to push into higher and higher resolution lineage tracing, I think, something that I really want to do in my own lab is really show all of the validation. I mean, really validate everything as much as we possibly can. I think this has been like a really good sort of norm of this DNA recording field. So I think keeping track of cells under microscopy in order to establish the ground truth before we sort of move on into situations where, of course, we're doing a new experiment, so it wasn't possible to keep track of the lineage any other way. And also these sorts of experiments where we validate the technology by knowing the relationships between populations in vitro.
Okay, any other question?
I have a question.
Sure, go ahead.
So you've been doing all this work in mammalian cells, and I'm curious if somebody wanted to do this sort of thing in, say, yeast cells or bacteria, would it even be possible? I mean, what sort of changes would have to be made? Do you have any idea about that?
Yeah. So the chiron work, I think it's an open question whether TDT would I think it's an open question whether it would work in yeast, because TDT is recruited to double strand breaks via its it's one of the first. So there's this really beautiful interplay in double-strand break repair where different repair proteins have like a different priority in terms of when they get a shot at the break. So we think TDT and its other sort of polymerase family members, its other family members have a very early shot, and we think TDT is likely in that group. And that's mediated by its interaction with the Ku complex, which is one of the earliest proteins to bind the double-strand break. So TDT is a sort of vertebrate innovation. So it definitely, like, it's definitely much more likely to match the Ku complexes of vertebrates. However, the Ku complex is conserved to yeast. So it's possible that either it would just work, or that by some engineering of the Ku complex that would retain its function, you could get it to work. For prime editor, chiron, I mean, I think it would work. I think it would work the same in yeast or bacteria. Insofar, I'm trying to remember if someone's published prime editor in those systems directly, but I think they have, at least in yeast. And there's not, it's kind of self-contained, like it doesn't require the exact interaction with the host machinery. One thing that does require is the way that the mutations are made is that, you know, you sort of have these two flaps, like the original DNA and then the new transcript, and you have to like resolve the flap. So that could possibly be different, especially in bacteria, which obviously have like a very different DNA repair setup. And in bacteria, I think I would be interested, but I think that CRISPR integrases might be a better way to go for sort of translating a nucleic acid that you can make into a durable record. But I mean, that's much more challenging in mammalian cells.
So you mentioned at the end of your talk that you're working on moving from whatever cell type you were using, I forgot, into other mammalian cell types. And I guess, are you running into issues with that? Or I mean, how different are these different cell types? They generally all work.
So the main source, so of course, I think most people in synthetic biology maybe start out in 293 T cells because they're so transfectable and they're so, but there's always the worry that they're just much more tolerant of doing weird stuff. So I have tested. So the thing I was most worried about is that the flap resolution would be just fundamentally different in different cell types. And especially that maybe the cells that grow well in culture are, you know, a bit more okay with, or, you know, maybe are so proliferative that they have lots of the flap endonuclease, and so it goes differently. However, I actually was able, early on, I did an experiment where I just, you know, made one of these moderately sized insertions in human primary cells, and I was actually able to get quite a good efficiency of that, you know, comparable with, you know, on the same order as base editing. So I think the fundamental mechanism is going to be conserved throughout mammalian cells. So I actually am not that worried, and we haven't had any like negative results yet. It's just a matter of, you know, I'm concerned about unknown unknowns always. And we're hoping to get those results really soon. In fact, I actually have cells that I nucleoaffected with the full PE chiron machinery that we're going to integrate by piggyback transposase. And they're, you know, they were alive yesterday, hopefully.
Wonderful. So I guess, I mean, your second paper will be in another, you know, same journal, or I mean, unless you wanted to go higher, Nature or Nature Biotech. Anyway, so we have another question. I want to, you know, quickly, you know, address this one as well, because this is from Sivang Josh. Thanks for the great talk and awesome work. How does the off-target nickase activity compare between these two system, original and the new one? Does this affect cell physiology, fitness? Also, what is your take on the recent nuclease tools coming from the Zhang lab?
Yeah. So I'm very excited about many of the recent nuclease tools that came out, from the Zhang lab and also from some other groups that now have these very small Cas9s whose exact cut geometry might be very advantageous for, first of all, the smaller it is. So, currently chiron has to be delivered in 2 viruses, and it would be vastly better if we had a smaller Cas9 that we could deliver in 1 virus just by itself. But then also the geometry might actually be quite a bit better for the purposes of recording. So yes, I think that those are definitely going to be really valuable for DNA recording, in addition to, of course, curing diseases and all of this really broad application. So in terms of the off targets, the off targets for chiron are potentially very significant. So we were able to do accurate lineage tracing, which I couldn't show you, but it's in the paper, in primary cells. So we know at least over a relatively short period of time, cells can tolerate both the repeated double-strand breaks, which, as I said, is a thing that happens in cells naturally, and also TDT potentially coming in not just to our double-strand break, but to potentially other double-strand breaks, and potentially making those a little bit more mutagenic. Although again, like TDT is a human protein that's actually expressed in human cells. So in specific human cell types. So there are definitely cells that can tolerate that machinery, but yes, there are probably significant off targets. Before we submit the paper, I would really like to fully characterize the PE chiron off targets. I think those are going to depend a lot on the exact targeting sequence that we use. So that's something we have full control over, as long as we optimize it. And so that's something that moving forward, I really want to, be very careful about. I do think that the likely consequences of PE chiron off targets are likely to be much less, because a nick is vastly easier to repair correctly than a double strand break. And unless you have, so the pegRNA requires a significant downstream sequence that matches the locus you're targeting. So unless you have that, it's highly unlikely that you'll actually get a mutation as a result of prime editor. So I think there are many reasons to think that it will be much better, but I think characterizing them, and also, as you said, just right now, as soon as we get our fully modified stable non-293T cell lines, we're gonna, keep track of their death and growth, and try to make sure that PE chiron isn't imposing a significant cost on the physiology of the cells.
Okay, so thank you. So actually, we are now 1 o'clock mark, and then actually I want to ask my question, but probably after this one. So thanks again all for attending. Next week, we will have Professor Ian Paulson from Sydney, basically 1 o'clock in the morning his time, and Bo Wang from NREL. And see you all next week at the same day and time as today. And then we'll have a follow-up discussion, as I said earlier, shortly with the Zoom link shown in the chat box. So see you in a minute. Thank you.